On Monday, we left off talking about red blood cells. We're not quite done talking about red blood cells. But as you guys remember, red blood cells were the oxygen and carbon dioxide carrying cells of our blood. And that is because of this chemical called hemoglobin that is found within our red blood cells. Okay. So what we're going to do now is talk about the life cycle of red blood cells and the recycling of hemoglobin when red blood cells die off. So reading through, life cycle of a red blood cell is only about 120 days because of the wear and tear the plasma membranes undergo as they squeeze through blood capillaries. Okay. Blood capillaries are about the same width as a red blood cell. Okay, and our blood is loaded with millions upon millions of these guys. So they're all moving through these same very tight windows at the same time. It'd be like if we all tried to walk through the door to leave this classroom at the same time. What's going to happen? We're going to get bunched up. We're going to get beat up a little bit. Okay. The other day we learned that red blood cells lack something. Irvin. They lack nuclei. Okay. They lack the nucleus because remember when they're developing, the nucleus is ejected, which enables them to store more hemoglobin. They also lack a lot of those other organelles. Okay. So when that red blood cell gets beat up as it passes through capillaries, it has no ability to repair itself. Okay. And so as they age, they get real fragile and start to break down or even burst. And so we have macrophages. We'll talk more about macrophages here in just a little bit. That are found in our spleen and liver that will take those and remove them from our blood circulation altogether. Okay. Now they don't just completely waste away the hemoglobin that's found in those red blood cells, they recycle it. Okay. Remember, hemoglobin consists of a heme, which is the non-protein parts. Okay. There's at the center of each heme an iron. Okay. That's going to be pulled away from the rest of the stuff. Okay. So the iron is removed and is recycled. Okay. Some of which will end up in our bone. Okay. And that way we can use it again for erythropoiesis. The other part of the heme, the non-iron parts, will get lost in our waste. Okay. And that's what causes your urine to look yellow sometimes and your feces to look brown. They are pigments. We have urobilin, which is where the urine yellow color of urine comes from. And then stercobilin, which is where the brown color of feces comes from. So if you ever wondered where, now you know. The other part of the protein, or the other part of the hemoglobin was the protein part, which we know proteins are made up of amino acids, so we can break those down, and then those can be used to make new proteins. Okay. Maybe even a new hemoglobin. The figure is pretty complex, and rightfully so, because it's a pretty complex process, but I tried to make it as simple as possible here at the top. Make sense? All right, let's now talk about concentrations of red blood cells and hemoglobin, something that are done to check on the state of the body and state of our blood. There are three common methods of reporting red blood cell and hemoglobin concentration. The first and probably the most common way is by measuring it with a hematocrit, which is where you take a sample of blood, the whole blood that is, so everything from it, you just pull it out, put it into a sample, and you find out how much, and it's percentage-wise, are red blood cells. Okay. If you see here, men are a little bit higher in red blood cell concentration than females. Okay. 40 to 54% in men, 38 to 46% in women, and that has to do with testosterone. That helps stimulate the production of erythropoiesis, which we're going to talk about erythropoiesis here on the next page. Okay. Letters A, B, and C show you three different blood samples that have been centrifuged out. Remember, a centrifuge is a machine that spins real fast and will separate things out. The really dense red blood cells will separate to the bottom. The very less dense or low dense plasma to the top. And then in the center between them will be this thing that we call Buffy coat, where you find the white blood cells and platelets. Okay. Anybody recognize which one of those would be considered normal? You would be wrong, actually. Letter A. Okay. Right about 40% of the sample is red blood cells. So letter A is normal. Letter B, we would actually consider to be anemic. Letter C, we would call polycythemic. We'll talk about anemia and polycythemia at the end of the lecture. Hemoglobin concentration is the second way, and this is specifically, obviously, measuring hemoglobin. Okay. We always want hemoglobin to be about 15 grams per deciliter. That's what we would say is normal. So between 12 to 18 is what they say the range should be. The more, the better, actually, because then you can carry more oxygen. But 15 is normal. And then just taking account of red blood cells per volume. Okay, this one's a lot trickier because you actually have to count them out. And you wouldn't take a deciliter of blood. You'd only take a microliter. And there's still going to be millions upon millions of them in that small, very small sample. All right, the other day we learned about the production of blood cells, and we described that as hemopoiesis or hematopoiesis. Both of those terms ended the same way, with poiesis. Okay. Notice this process, which is specific to red blood cells, is called erythropoiesis, which, remember, erythrocytes are the fancy way to describe, or is the fancy way to describe red blood cells. Okay. So I haven't really looked it up in a while, but I'm pretty sure if I remember correctly, poiesis just is a word or suffix that means formation or forming. Okay. So erythropoiesis pretty much translates to red blood cell formation. It's a negative feedback mechanism that begins with a low oxygen count or level in our blood. Okay. Measured in our kidneys, actually, uniquely enough, because our kidneys actually are the structures that filter our blood, and a lot of our blood at rest goes to our kidneys. Okay. Low levels of oxygen in our blood are called hypoxia. Okay. So if we detect hypoxia in our kidneys, okay, we will secrete the hormone erythropoietin, EPO. EPO is the hormone that produces or causes erythropoiesis. Okay. Now, erythropoiesis, the actual process, involves precursor cells in our bone marrow called proerythroblasts. Okay. Proerythroblasts differentiate through cell divisions to become reticulocytes. Now, this reticulocyte here looks just like a red blood cell, but it's not quite to that point. What we see the reticulocyte is that it has the same shape as red blood cells because it's ejected its nucleus. Okay. And we saw that in that figure a couple of slides ago back on Monday. Okay. And that's where that distinctive shape comes from. The reticulocytes are actually what leave our bone marrow, entering into our capillaries, and then a couple of days later actually become the fully functioning red blood cells. Now, if there's more red blood cells, there's more ability to carry oxygen, and so if there's more oxygen, therefore there's better delivery to our kidneys. Therefore, the feedback loop would shut down, demonstrating that this is negative feedback. And things that can stimulate or even simulate hypoxia. Okay. Obviously, high altitude can lead to hypoxia in our body. Okay. Because there's not less oxygen necessarily in the air, but there's less oxygen drive into our body. So it's harder to get the oxygen into our blood. Okay. Exercise actually can stimulate it. Okay. Because when we exercise, we actually shut down blood flow to the kidneys, and therefore our kidneys kind of interpret that as having less blood. Okay. And so prolonged exercise can actually promote a little bit of EPO release. And then loss of lung tissue or function obviously means we can't get in as much oxygen into our blood, therefore being the same essentially process as what happens when we are at high altitude. All right, that's it for red blood cells. We'll now move on to our second type of formed elements, which are the white blood cells or leukocytes. These are the protectors of our blood, protecting us against infectious microorganisms and other pathogens. They're different than red blood cells in that they retain those organelles, which is important because it helps us categorize them. There are two major categories that we fit leukocytes into: granulocytes and agranulocytes. And it has everything to do with what they look like when you pull them out of the blood sample and stain them and look at them under a microscope. The granulocytes appear grainy when they are stained because of lysosomes and other membrane-bound organelles that really accept the stain. These all end with the suffix -phil. Okay, so you have eosinophils, basophils, and neutrophils. Eosinophils combat the effects of histamine in allergic reactions, kind of similar to what antihistamines do for us when we ingest those to, combat our allergies. Some of you guys are probably experiencing that now as we're starting to warm up. Phagocytized antigen-antibody complexes, which we're going to talk about antigen-antibody complexes in a little bit. Remember, phagocytosis from earlier in the semester is where cells will chew stuff up, literally cellular eating, okay? And they destroy some certain parasitic worms. Okay? These cells actually have the capacity to leave capillaries and enter tissue fluid, which is pretty cool. And most of your white blood cells actually do also share that same property. We'll talk about that process in just a bit. Basophils liberate heparin, histamine, serotonin, involved in allergic reactions that intensify inflammation or the inflammatory response. We'll talk more about the inflammatory response in our next lecture, so that will be after break. Okay. These guys are actually similar to mast cells. I mentioned mast cells just a little bit the other day, that they both release substances involved in inflammation. And then neutrophils, something that's important about these is that they're the first responders. Okay, so they're the first white blood cells that arrive at an infection site. And these are one of the two major types of phagocytes. Okay. And we'll see the other major phagocyte on the next page. Okay. After engulfing a pathogen, neutrophils unleash several chemicals. Lysozyme, which is an antibacterial enzyme. Oxidants are kind of like bleach, in that they can kill just about anything. And then defensins, we'll talk a little bit about those. We'll talk more about those chemicals in our next lecture, which really, as you can tell, relates to the blood. And that's why I like to talk about it right after we've talked about the blood. The other type of white blood cells are the cytes, or as we see here, the cytes. I call them the cytes because that's what they end in. You have lymphocytes, which are the major soldiers of the immune system. Okay. Our immune responses, many of those responses anyway, take place within our lymphatic organs, like our lymph nodes, which circulate or are part of the lymphatic circulation, which is where you find lymph. Hopefully you guys are seeing a trend here with the word lymph, either the actual word lymph or the beginning of the word like lymphocytes, or lymphatic, or lymph nodes. Okay. So these are important cells, and we'll talk more about these in our next lecture. Okay. You actually find several different sizes. Not that that's super important physiologically that we are aware of. It's actually pretty useful because we know, like, for example, a large number of lymphocytes that are large will increase in viral infections. So it helps with diagnoses or diagnostics. The two major types that we'll talk about are T's and B cells, or T lymphocytes and B lymphocytes. There's a third type, natural killer cells. I mentioned those a little bit the other day. But T lymphocytes attack viruses, fungi, transplanted cells, cancer cells, some bacteria. Since they attack a transplanted cell or a transplanted organ, they can be really bad for us. If we have a heart transplant or something like that, we have to make sure these guys don't work at their max capacity, because they can reject that organ, and they can also reject transfusions. Okay, so that's not necessarily a good thing. Okay. B lymphocytes mature and develop in our bone marrow, whereas T lymphocytes, we call them T's because they develop in our thymus. Okay. But B cells are particularly effective in destroying bacteria and inactivating their toxins. Natural killer cells are a little bit more just kind of, they go after just about anything. So they go after a wide variety of infectious microbes and certain spontaneously arising tumor cells. The other type of phagocytes, or the other major type of phagocytes, are macrophages, which form from monocytes that have left the blood. Okay. It might not be a bad idea. I know it's not written here. But neutrophils and macrophages are your major phagocytes. So it might not be a bad idea to write that down. Two major phagocytes, macrophages and neutrophils. Neutrophils, we just talked about, were the first responders. Macrophages come next. They actually come in much larger numbers, though. So they're like the second wave and the much stronger wave. And there are two types of macrophages: fixed, which stay where they're at, and wandering, which you can tell by their name, wander or roam around. So all those white blood cells have a specific role, and many of them target specific things or go to areas of the body that are in a certain situation, like a damage or a cut or a bacterial invasion. Well, how do they know where to go? Okay. Well, there's a pretty cool process, and I have a pretty cool analogy to describe that, and I'll get to that here in a second. So let's read through. Skin and mucous membranes of the body are continuously exposed to microbes and their toxins. Some of these are able to invade deeper tissues to cause diseases. In order to combat these microbes and toxins, white blood cells will leave the bloodstream, collecting at sites of pathogen invasion or inflammation. And that's what we're showing here. Okay. Now, the inside of your blood vessels, in a healthy blood vessel anyway, should be like a non-stick pan. Okay. Which are pans that obviously don't allow sticking. Okay? So even if you're burning something, hopefully you can still wash it out and it's still going to be just like it is. The reason our blood vessels are non-stick is because they have a prostacyclin coating. It's like putting PAM on it, or that non-stick coating on your pans. Okay? But when blood vessels get damaged, they can see, or places can, let me just back up. Okay, I'm getting ahead of myself. When we want white blood cells to leave, they can actually stick to that non-stick coating, and then they can emigrate out by sticking themselves between the cells that form the blood vessel. Okay. You can see here a singular blood vessel that we've opened up to see inside. Do you notice that these little lines here, those are the plasma membranes of the cells that form it. Okay? So these guys are not completely fluid tight. They're predominantly fluid tight, but they do allow sometimes small things like white blood cells to pull themselves through. That process is called emigration. Okay? Now our neutrophils and macrophages, those two primary phagocytes, have a really innate ability to do this. Okay? And then they can perform phagocytosis, which is where they ingest bacteria and dispose of the dead matter. Now, how do they know to get there? Well, this is where my analogy comes into play. Okay. My wife is a fantastic baker. She loves to make cookies and treats and all sorts of other wonderful things. And I say they're wonderful because I have a sweet tooth, unfortunately. But when she's making brownies especially, that seems to always waft throughout the house. So if I'm sitting downstairs watching a game or something and she's upstairs making brownies, there's a good chance I'm going to start to smell those brownies getting cooked or baked. And what am I going to do every time? I'm going to walk myself up the stairs and I'm going to try to give me some brownies or some brownie batter. Okay. I have that chemical attraction to the kitchen. Okay. White blood cells have a chemical attraction to areas of trauma because damaged cells and even bacteria will release chemicals into the blood. And so they kind of smell it out or sniff it out and know where to go. So it's pretty cool how that works. That process is called chemotaxis. Okay. Now, I had mentioned a second ago that we're going to talk more about lysozymes, defensins, and oxidants. Those are chemicals that help those phagocytes digest and dispose of those viruses and bacteria and that kind of stuff. We'll talk more about those in the next lecture. Before we move on from white blood cells, We'll talk about this concept called white blood cell counts. We learned the other day that red blood cells and platelets like to stay steady in their number. Well, white blood cells, we learned, don't, because those levels will go up and down. They'll fluctuate. Okay? Any increase in white blood cell tells the body something's wrong, or is because something in the body is wrong, and that usually is indicative of inflammation or infection. We call this leukocytosis. Things like invading microbes, strenuous exercise, anesthesia, and surgery promote leukocytosis. Now, this is a benefit to doctors because we know, for example, that neutrophils increase in bacterial infections. And so, they might order a differential count or differential white blood count to find out what white blood cells are elevated. And if they see neutrophils are elevated, they might be able to say, hey, it's probably a bacteria. Anybody in here like to watch the show House? If you don't, and you're interested in medicine or health, which I know probably a lot of you are since you're taking this class, I'd say give it a shot. It's a pretty good show. Hey, if you're not familiar with it, Dr. House is the best diagnostic medicine doctor in this hospital, and his job is to figure out what's wrong with people that nobody else can figure out. So what he does is gets a team together, and they put a list of the signs and symptoms. Okay? And then pretty much every episode you hear him say, well, let's get a differential on. Okay? What they're doing is drawing blood, and then they're going to try to find out what white blood cells are elevated. That way they can figure out if it's a virus, if it's a fungus, if it's a bacteria, if it's a toxin. Okay? And so that's pretty cool, kind of as a thing that you can kind of apply to what you're learning now to a pretty neat show. Okay. Now, the opposite of leukocytosis is leukopenia, which is where white blood cell levels are low. Never is a good thing. Okay. Radiation, shock, chemotherapy can cause leukopenia, which means that the immune system can become compromised. Therefore, you are at great risk for disease. All right, table's not that important. It's kind of cool to see what numbers go up and down based on what's happening in the body. All right, that's it for white blood cells. We'll now move on to platelets. Platelets are actually not actually cells. They're small fragments of cells called megakaryocytes. The production of platelets is through a process called thrombopoiesis, and that's due to a hormone called thrombopoietin. So we saw just a little bit ago erythropoietin and erythropoiesis. Now we get to see thrombopoietin and thrombopoiesis. It begins in the bone marrow with myeloid stem cells that become megakaryocytes. And these megakaryocytes grow and then start to divide off the plasma membrane and the cytosol. And then what happens is those little divisions start to break free, and that's what forms the platelets. The platelets then enter the bloodstream, and they'll stay only for about a week before they become damaged and worn out, where they will then have to be removed by those macrophages that we've talked about. When they are in our bloodstream, they do a heck of a lot of things, as you can see here. Okay. One of the most important things they do is help us stop bleeding when we have a cut. Cut to a blood vessel, so when a blood vessel is broken, they will secrete vasoconstrictors. Now, vasoconstriction shuts down blood flow in a blood vessel. So like kinking a garden hose, which is what we want to do if we're leaking from that blood vessel. We don't want blood spilling out, so we shut down blood into that blood vessel. Okay. They also come together and form what we call platelet plugs, which I like to think of like putting a piece of bubble gum on a leaking pipe. Is that a long-term solution? No, but it might get the job done in the short term. Okay. And we'll talk about platelet plugs here on the next slide. They also secrete procoagulants. The word coagulation is a fancy word to describe clotting. Okay. Pro means for, so they are for coagulation. as it says there, promotes blood clotting. They also help to dissolve blood clots. Blood clots that have outlived their usefulness are bad. So we don't want them hanging out in the blood. I'll talk a little bit more about that here in a minute. They secrete chemicals attracting neutrophils and monocytes, the sites of inflammation. That's that chemotaxis I was talking about. They can actually phagocytize and destroy bacteria, which is kind of cool. And they also secrete growth factors that stimulate mitosis in fibroblasts and smooth muscle in order to help regenerate that broken blood vessel. So they help repair. All right. Table 19.3 is just a nice summary table from your textbook of those various 3 different formed elements. Blood, red blood cells, white blood cells, platelets. It even gives them numbers, characteristics, functions. I just threw it in there because it was in your book. Let's now talk about hemostasis, which is the sequence of responses that ultimately will stop bleeding. Okay. And this happens when blood vessels first get damaged and blood starts spilling out. Sometimes we see it as it's coming out of our skin. Sometimes it's internal, and we don't see it, but can be very dangerous. Okay. This response must be very quick, must be localized to the region of damage, and carefully controlled in order to be effective. The first thing we're going to do is something I just talked about. We're immediately going to shut down blood flow through that, or we're going to diminish the amount of blood flow through that broken blood vessel. That's this vascular spasm. So this prompt vasoconstriction or tightening of a broken blood vessel to prevent blood loss. Okay. How it works is tissue damage can stimulate the smooth muscle of the vascular bed, so all those blood vessels in an attempt to decrease blood flow. So it causes the muscles to go like this. Okay. It pinches them off. Okay. This effect can last for several minutes to several hours. Over here on the right, we've got 4 figures. The first 3, you can see that are numbered, 1, 2, and 3, are showing a platelet plug formation. Okay. That's that piece of bubble gum covering our leaky pipe. Okay. As I mentioned earlier, healthy blood vessels are like a non-stick pan. But when they get broken, the collagen fibers that are found within the blood vessel will stick out. And those are places where platelets can stick. Okay. And so that's what we're showing up here with step 1, platelet adhesion. Okay. So those collagen fibers allow for anchoring points for platelets, and the mass of platelets that initially forms is called the platelet plug, which we see finally forming in step 3. How that works is you need to recruit more platelets to that area, and that's done due to 3 different chemicals that are released. As the platelets aggregate, they release several chemicals that help to promote further formation of the platelet plug. Adenosine diphosphate, serotonin, and thromboxane A2. Now, when you put a piece of bubble gum over a leaky pipe, is that a long-term solution? No. So that's going to be the step before the actual blood clot's formed. Coagulation is the actual process. It's called clotting, or more commonly known as clotting, which is where we convert fibrinogen, that plasma protein we talked about the other day, into fibrin. Fibrin is a sticky protein adhering to the walls of the vessel, and that's where a mass of cells and platelets seals off that broken blood vessel, and no more blood should be able to spill out. And then over time, the blood vessel will repair itself. And so, pretty cool process. Let's talk more about coagulation, because it's a pretty complex process involving many steps and conversions of certain different proteins. It involves substances called clotting factors. Calcium, inactive enzymes. Those enzymes formed by hepatocytes in our liver, released in the blood, and then other various molecules associated with platelets or released by damaged tissues. Okay. This is actually an example of positive feedback that you can think of kind of like a domino effect, where one domino knocks down the next domino and next knocks down the next, on and on and on and on. Okay. So it's a complex cascade of enzymatic reactions in which each clotting factor activates many molecules of the next one in a fixed sequence. There are two pathways, which we have labeled over here in our figure, an extrinsic and an intrinsic pathway, or as we call mechanisms. Okay. The extrinsic is initiated by clotting factors released by either the damaged blood vessels themselves or what we call perivascular tissue, which is tissue located outside of the blood vessel and in close proximity to it. Okay. So extrinsic you can think of as outside of the blood, whereas intrinsic is within or in direct contact with the blood. Okay, that's where the word intrinsic is coming from, in or within contact with the blood. Now, it doesn't matter if it's extrinsic or intrinsic, because guess what? The end result is still the same, which is eventually leading to the formation of prothrombinase. Prothrombinase is an enzyme converts prothrombin to thrombin, which we can see here is step 2 in our figure. Okay. Thrombin is the enzyme that then takes the fibrinogen, chopping it up into fibrin, which will then cause the mass of cells and platelets and all that stuff to form the clot. And that's how we form a clot. Doesn't matter how we get there, the end result is still the same. Now, unfortunately, sometimes conditions can cause inappropriate clotting or clotting at the incorrect time. If the healthy blood vessel, or if a blood vessel is in a healthy state, it's going to exhibit this platelet repulsion that I've been talking about. It's going to have that non-stick coating from the prostacyclin that I was talking about earlier. Okay, an example of disorder that illustrates an inability to have platelet repulsion is atherosclerosis. Look at our two figures at the bottom. These are actually blood vessels zoomed in 20 times in a light microscope. In a healthy blood vessel, you can see the lumen's very wide, nothing is stuck to it. Versus in an atherosclerotic state, plaque builds up and narrows off or occludes, as we like to say, the lumen, and therefore blood flow is partially obstructed. Okay. Atherosclerosis is sometimes just called fatty blood vessels, okay, which can be dangerous because it can cause complete occlusion to blood flow. Very dangerous if that's in your heart or if it's in your brain. Those are the things that cause heart attacks and strokes. Okay. Now, we just learned thrombosis is the fancy way to describe clotting, okay, which the other word to describe is coagulation. Okay. So here in a second we're going to learn that thrombins are, or a thrombus is a fancy word for a clot. The word antithrombin is any substances or any substance that delays, suppresses, or prevents blood clots by inactivating thrombins or thrombin, and thus prevents thrombin from converting fibrinogen into fibrin. Okay. Sometimes you'll hear these called just blood thinners. Okay. It could be as simple as aspirin that I mentioned the other day. It could be as complex as heparin. Heparin is a chemical. It's used as a drug. Okay. If you watch House or if you're in a medical setting, there's a good chance you're going to hear the word heparin given to patients in many, many cases. It's more of a complex blood thinner, which is not actually a blood thinner. It just prevents clotting. I'm sure most of you guys have heard of hemophilia before, right? Hemophilia is a disease, it's a genetic disease, where a person has an inability to clot their blood. Okay, so they are deficient in clotting factor. Any of those clotting factors. Okay, so any simple small cut can be very dangerous for a person with hemophilia. Great thing is, though, it's a very rare disease, anymore anyway. It's a disease that's actually mostly found in men, for those that actually have it. It's typically found in men because they inherit it from the X chromosome. And who do they get the X chromosome from, mom or dad? Mom. Okay. And for mom, she most likely won't have the disease. It will never know that she's even a carrier for it. Because for females to have hemophilia, they have to have the allele on both of their X's. Because females have XX, Men only have XY. So mom can be a carrier and not affected at all, but then if she's having a boy, there's a 50% chance that that's going to be containing hemophilia. Very common, actually, a long time ago. I think it was a Russian royal family, which happens when you don't marry outside of your family. So they had siblings and cousins getting married and making babies. And small diseases or rare diseases like this can be spread much easier. Thrombosis, abnormal clotting of blood in a broken blood vessel, something that's very dangerous, especially if you're having like a surgery. That's why they put TED hose on people and that kind of stuff, keep their circulation correctly occurring. I mentioned just a second ago the word thrombus is a word that means clots, which can grow very large and obstruct blood flow. You can see that here. If it breaks free, it can travel in our bloodstream as what we call an embolus, which can then lodge itself somewhere else, blocking blood flow. Very, very dangerous. This is, sometimes what happens after surgeries and that kind of stuff. Just kind of a freak thing. Derek Thomas was a Chiefs player back in the 90s, died of a blood clot after he had broken his, I think it was his back, if I'm remembering correctly. There's really not much they can do if it happens. Anybody ever heard of pulmonary embolism? Yeah, that's what they're talking about. Pulmonary refers to when it happens in the lungs. All right, what I want to do now, though, is switch over from clotting to talking about blood typing, something that involves our red blood cells. Okay, something that's important. You guys are probably aware of what your blood type is. It's not only important just to know, but it also affects transfusion compatibility. And it's specific to the plasma proteins and the proteins found on those erythrocytes. Okay. Now, we're going to talk a lot more about antigens in our next lecture. The word antigen refers to complex molecules found on the surface of cells. Our cells have antigens, as do bacteria and viruses. Okay. We use the word antigen when we talk about immune responses as something that our body is going to attack. But our cells also have antigens, okay, which are going to be proteins, glycoprotein, glycolipid structures. These are going to be very unique. They're genetically unique to each individual. They'll be found on the surface of the cell, as I just mentioned. And why we talk about those when we get to the immune system is because that's how our immune system is able to invade or to attack an invader. Okay, because it knows that it's not supposed to be within it. Okay. We'll also talk more about antibodies. And I already mentioned a little bit about antibodies, but these are proteins that are dissolved in our blood plasma that will attack, in a kind of sort of way, those antigens and mark them for destruction, like what we see down here with antibodies binding to antigens on the red blood cell, which actually is not a good thing. Okay. Because that would cause this to occur, which is what we call agglutination, okay, where antibody molecules bind two or more antigens to form a large antigen-antibody complex. This occurs if you receive the wrong type of blood in a transfusion, okay, like a person with type B blood receiving type A blood. Okay. Look what happens to the red blood cells. They're clumping up. Okay. That's what agglutination is. Okay. And it has everything to do with the agglutinogens and the agglutinins, which are the fancy ways to describe the antigens on the red blood cells and the antibodies that are found in the plasma. Okay. Let's talk about those antigens and antibodies. Those determine your blood type, which we classify into one of four types: A, B, AB, or O. Okay. We name the blood type based on what antigen, or as I said on the previous page, agglutinogen your red blood cell has. So what I mean is type A blood is named because it has A antigen. Type B blood has B antigen. AB blood has A and B. And I always tell students, if it helps, think of type O as in like type 0. like no antigens. Okay. Now, in a healthy blood, the antigens and antibodies are going to be the opposite to prevent them from clumping up. So antigen A in type A is always going to be paired with antibody B. So type B with antigen B has antibody A. If they have both antigens as in AB, they can't have any antibodies. And then if they have type O with none, they can then both have both antibodies. Okay. So no, plasma never contains antibodies binding to antibodies on its own cells. That should say antigens on its own cells, not antibodies. Okay. That was a typo on my part. So let's look at what happens when people receive different types of blood. Okay. We've got a kind of matrix graph or table over here on the left, and a figure on the right that I'll talk about in just a second. But in the blue shaded columns, we've got recipient's blood with breakdown on antigens and antibodies, which we just talked about. And then what happens to their blood when they receive a donor, from a donor, I should say, or donor blood. Okay. One thing that's always going to occur is you can always receive your own type of blood. Type A can always receive A, type B can always receive B. AB can always receive AB, O can always receive O. Okay. You can also always receive O, because O is the universal donor. Okay. If you look here, donor type O, no agglutination occurring in any of the recipients. Okay. O, unfortunately, can only receive O, okay, because if it's type O, it agglutinates for the other three types. Okay. That's because of those antibodies that are found in it. Okay. Type AB, as you can see, can receive from everybody. That's why we call it the universal recipient. Okay. Now, this is a figure on the right side of the page, or I guess it's the right side of the page, that describes and shows an experiment to actually do blood typing. If we actually had a physical lab in this class, we would do this. It's pretty fun, it's pretty cool. I've done it both with actually taking samples of blood by doing a finger prick. And I've also done it with synthetic blood. They make these sera that have antibodies in them. Okay? And you can take a drop of blood, whether it's synthetic or real, and then take a drop of anti-A serum and anti-B serum, drop it on there, and if it clumps up, as you can see here, it tells you that's what antigen is found on it. So if we didn't know that this is type B, and it clumped up when it got antibody B, it tells you right away there's antigen B on that blood. And for AB, you can see it clumps up with both A and B. That tells you it's positive for A and B antigens, so it has to be AB blood. And then like type O, doesn't agglutinate on any of it. Okay. Now, when we talk about blood typing, there's one thing else that we generally add after A and B and O. What is it? Positive and negative. And that is due to another antigen, what we call the Rh factor. Okay. If they are positive, it means they have another antigen, the Rh factor. Okay. So the Rh antigen is present on the red blood cell. If they're negative, doesn't mean a bad thing. Okay, I grew up thinking it was bad because I had negative blood. I was freaked out about it as a kid. Okay, it just means that I didn't have the Rh antigen. Okay. Now, is there an Rh antibody? In a normal sense, no. Okay. Normally, blood plasma doesn't contain the anti-Rh antibodies. But if a person with Rh negative blood receives Rh positive blood, then they're going to start making it. Okay. If they receive a second transfusion with Rh positive blood, those antibodies they made are going to attack those new red blood cells, therefore destroying them in a process called hemolysis, which is where the red blood cells are like a broken water balloon. They pop. Okay. Over there on the table on the right, it gives you a nice breakdown of the 8 different blood types when you combine positive, negative, and the 4 A's, B's, O's, and ABs. It gives you antigens, it gives you antibodies. Notice how they put RH, RH, RH for the negatives with a question mark behind it. Typically, it would be no, but if they've had a transfusion, yes, possibly. And then it even gives you the percent of the population. Percent of the population isn't that important to me, but the other stuff is. There will definitely be questions on the test related to the other stuff. I will ask you on the test, a person with AB blood has which antigens? Or if they're AB positive, what antibodies do they have? Etc., etc. There will be multiple questions related to blood typing. Another issue can occur when mom is Rh negative, but her baby is Rh positive. That is possible because your Rh factor is determined by both mom and dad's genetics. So she is capable of carrying a positive baby when she's negative. Okay? When it becomes an issue is if during labor a little bit of bleeding occurs with the placenta. Okay? And then mom might start to make anti-Rh antibodies unless treated with a RhoGAM shot shortly after birth. RhoGAM also given during cases of miscarriage and abortion as a precaution. Why it's an issue is not because of her, but if she has another baby that's Rh positive. Because those antibodies will attack the fetus and it can cause hemolysis. That's why they call it hemolytic disease of the newborn. Those blood cells will burst, can cause brain damage and even death. So very dangerous. Now it says here about extremely important to know both donor and recipient before blood is transfused to avoid transfusion reaction. It's also important to know the type of mom and baby. And they're going to find that out. Obviously mom really early in pregnancy, but a baby right off the bat. So. All right, last thing I want to talk about are blood disorders. Okay, I'm sure most of you guys have heard of anemia before. Anybody not ever heard of anemia before? How many of you guys knew that it wasn't actually being deficient in iron? Not necessarily, anyway. Most people believe it is deficiency of iron. The most common form of anemia is actually iron deficient anemia. But the technical definition of anemia is a condition in which oxygen carrying capacity of the blood is reduced. So you have an inability to carry oxygen in your blood, which can come from being deficient in iron. It can be because you are deficient in red blood cells. That figure I showed you guys a little bit ago that said where the red blood cells were really low, I said that was anemia. Okay. They're going to be deficient in their ability to carry oxygen because they don't have enough red blood cells. Okay. Women are especially at risk for iron deficient anemia. Okay, there's a lot of different reasons for it, but iron, as we know, is that component of hemoglobin, and specifically the part of the hemoglobin that carries the oxygen, and that's why being deficient in iron can lead to anemia. Okay. Can be due to inadequate absorption of iron, loss of iron, and increased iron requirement, or even just insufficient iron intake. Now, having a hemorrhage, bleeding a lot, can cause anemia. That's what hemorrhagic anemia is. There are several different forms of anemia. I just picked a couple of them. The opposite of anemia is polycythemia. High percentage of red blood cells in your whole blood. Okay. This raises the viscosity of blood or thickness of blood, making resistance of flow really high. Therefore, the pump of the blood, or the blood has to pump harder, and it's very difficult sometimes. Okay. It causes high blood pressure and can even lead to stroke or even a heart attack. Now, this can be caused by just abnormal increases in red blood cell production, hypoxia, which remember was the whole stimulus for the production of red blood cells, dehydration, which is where the fluid in the body is low and therefore the plasma is low and therefore red blood cells are too high in number compared to plasma, blood doping, or even artificial ingestion of EPO. Okay, both blood doping and EPO usage are illegal by most sports. Okay, blood doping, if you're not familiar with it, is where people take out a sample of blood, they store it, then they wait for their body to regenerate their blood over time, and then just before they get ready to perform in an activity, they stick that blood back into their body, which means they have more red blood cells, which means they can carry more oxygen, which means they can make more ATP. Therefore, and we know ATP is absolutely necessary for muscles to contract. So it's kind of a cool process, but it's very dangerous. Sickle cell disease, sometimes called sickle cell anemia because it leads to anemia. Disorder in which the red blood cells of an individual contain HbS, which is an abnormal kind of hemoglobin, which causes the red blood cells to bend into a sickle shape. Okay. Anybody know who's at risk, highest risk for sickle cell disease? African Americans. Okay. It actually is traced back to Africa, where most African Americans originate from. Okay. The HbS allele, because there's actually two different alleles for hemoglobin, HbA and HbS. If a person has HbA and HbS, they don't actually have anemia, or they don't have sickle cell disease, but they're malaria resistant. So it's actually a way to help prevent malaria, which is a disease very rampant in Africa. Leukemia, whole group of disorders, cancer disorders that is, in our bone marrow, causing white blood cells to multiply, and oftentimes forming tumors in our lymph nodes and other places in our body, like lymphoma, non-Hodgkin's, Hodgkin's, and all sorts of different types. I just included just the major category. All right, that's it.